Hello, everyone, and welcome. This is our weekly HEALEY ALS Platform Trial webinar, and I'm just giving people a few seconds to join from the waiting room. So thank you for joining, as always. Really great to see people attend this meeting regularly, and also thank you to all of you who are watching the recording of this webinar. As always, we post our recordings and slides on our website. This is Sabrina Paganoni. I'm speaking from the HEALEY Center at MGH. And as always, I'm here with our patient navigation team, Alison Bulat and Catherine Small. And tonight, it's really my honor to introduce our guest speaker, Dr. Jeffrey Rothstein from Johns Hopkins University. And the reason we invited him is really because of his really international expertise and leadership in several topics related to ALS, but in particular for tonight, we really wanted him to talk about the development of new science, new biomarkers, the importance of collecting new biomarkers as part of trials like the platform trial, so that together with him and his team of scientists and collaborators, we can continue to learn more about ALS, which is really one of the most important scientific goals of the trial. And so with that, Jeff, you have too many titles and accomplishments, so I'm not going to even try to summarize them, but maybe you can give us a brief summary and present your group and the work you're doing.
Sure.
Jeff?
Thanks, Sabrina. And thanks, everyone, for signing on. I'm going to go relatively fast, but I understand the slides are going to be available. And if anyone has questions, certainly contact me either through email, phone calls, doesn't matter. I love talking to people, especially in the evenings. So I'm going to do just very quickly a background to some of this, which has to do with the Packard Center. Next slide. I started the Packard Center back in 2000 as an academic consortium to bring academics together to understand what causes ALS, and developed really the tools so we can understand what causes ALS, but also to serve as a platform to discover drugs. And quite frankly, many of the drugs or pathways in ALS have come from these researchers. And let me be very clear, although I'm at Johns Hopkins, these are researchers, I'll show you a slide in just a second, from really all over the world. We cherry-picked the best junior and senior researchers to work together in a highly collaborative way. And this requires rapid funding. We can fund people within 2 months. We've done this now this is our 23rd year. And so this is just a great group of people. And every year we have a very large meeting. I know some people have actually signed on where we have about 300 to 400 researchers in person or on web who review the science that we're doing. So next slide. And this slide just summarizes, I'm really not going to go into it. If I look over the time period, we have an enormous number of researchers who've worked together over multiple continents, and we include their lab members. It's essentially one big academic pharma company, if you will, except we're not a company. And we've really developed, not me, but really developed most of the tools that we use today in ALS to understand the disease. Next slide. And this slide it's a little outdated, but it actually shows the breadth of researchers that have gotten together in this collaborative effort to understand ALS. And by the way, this doesn't mean that all the other organizations that participate in ALS research are not important. In fact, we often collaborate and share grants with ALSA and MDA and ALS Finding a Cure. So it's very common. Next slide. Sorry, I went that fast. One of the really neat accomplishments that we've worked on for the last 5 years is something known as the Answer ALS Research Platform. There is an Answer ALS Foundation. They actually funnel money to help pay for this. This is an enormous collaboration of 100 researchers around the United States to collect and follow over 1,000 ALS patients, where we collected clinical data and also then generated whole genome data from all our patients. But most importantly, we generated, and I'll show you, this is some of the new research in a moment, IPS lines, or induced pluripotent cell lines. These are stem cell lines that we generate from patients' blood today. We convert those cell lines into essentially their spinal cord, and it serves as biopsy. And from those tissue samples, if you will, we've then gone on to generate additional data about RNA and protein and DNA, and together it amounts to 6 billion data points per patient. All of the data, when I designed this program, all of the data is freely accessible to both academics, but also companies, because we need companies as part of this consortium to eventually develop drugs for ALS. That's also a web portal. It's now called NeuroMind that people can access with the appropriate permissions. Next slide. So that was just a really quick background. What I'm going to talk about is really the development today of new possible biomarkers for ALS. We all know about biomarkers, and in fact, just yesterday, the FDA reviewed the SOD1 gene therapy, and one of the highlights of that was they accepted that neurofilament, a protein that's presumably released from neurons when they die, is an acceptable result of changes in patients, and potentially drugs that change those levels that we can measure either in cerebrospinal fluid for this protein or in blood could be indicative of the drugs working well. These are important milestones as we approach ALS. Now, to be clear, not every drug may affect NFL, but it is an important biomarker. And this slide just actually summarizes a number of different biomarkers that we, the larger community, have been working on to help us better not just understand ALS, tell us if our drugs are working, and of course, tell us if the drugs are working at their target and at working at the clinical outcomes. By the way, when I use the word target, when we develop drugs, especially pharmaceutical companies, just because they made a drug doesn't mean it's working in the brain the way it should work. And that's an important marker of a drug working properly. It's called target engagement. And if a drug, for example, if a drug can't get into the brain and it's supposed to get into the brain to work, then that drug's really never going to work in patients. And today, people like Sabrina and I and others that are part of Neil's review with companies, how well have they developed their drugs? Do they have biomarkers of drug action? And this slide talks about MRI approaches, that is imaging approaches, PET scanning, positron emission tomography, very specialized approach, not really used extensively in trials, more for research. But most importantly, in the middle of that column, it's blood or CSF, cerebrospinal fluid, that comes from spinal taps, or even urine that may contain markers that we think could be important. The slide lists a number of them. They all have not been fully validated. These are things that we as a community are thinking of. The most common one we use today is something called neurofilament light chain. Not really going to talk about that because we use it. To be very clear, not all biomarkers are specific. NFL, neurofilament light chain, is seen in a large list of diseases, brain trauma, Alzheimer's disease, multiple sclerosis. It's not unique to ALS, but it elevates, it rises in ALS. And if a drug's protecting the nervous system, we think the drug may read out as lowering NFL. And that's exactly what happened in the tofersen study. By the way, not all drugs may do that. Drugs that work on muscle may have nothing to do with NFL, but most of our drugs that we developed for ALS today are really working at the brain and spinal cord to slow the disease down. And NFL is one marker. What I'm going to talk about today, let's go to the next slide, is a new biomarker. And I'll come to that in a minute. But when we think about ALS and biomarkers, we think of markers that apply to different stages of disease. One of the things we're really interested in developing, again, Royal We, is markers, and you see that blue line very early when there's actually no disease, before the disease shows itself. Can we start detecting the disease? This has already been studied for NFL. We know with patients who have the genetic forms of ALS, therefore we know they're going to get ALS one day, we can detect some of these biomarkers right before disease starts. And we're looking for more of these biomarkers because we don't really know if one's sufficient, 2 or 3 or 4. They tell us different stages of the disease. But these are the kinds of things that we're working on. In the more uncommon C9orf form of ALS, that is that gene mutation, there are aberrant proteins that are made. They're called DPRs, and that's shown on this yellow bar. And we know we can detect them very early in the disease. Even before disease begins, we can measure those proteins. Now, we also know that these markers don't always tell us about the severity of the disease. They may be markers that there's injury to the nervous system, but they don't always tell us how injured the nervous system is. These are the kinds of questions and many more. Does the biomarker correlate with the disease? Is it seen early in the disease? Does it go away with disease? As an example, the C9 gene therapy lowered that dipeptide protein very dramatically. It actually didn't correlate with any improvement in disease. So there's different roles for these biomarkers, and there's much that we have to learn about them. So you as a patient community will hear about us talking about them, but you have to keep in mind there's still much we don't yet know about them. But if we don't study them and learn more about them, we'll never know. We'll never know how useful they are for the most important part of the disease, that is drug, or one element that's very important, drug discovery. Next slide. The main reason I'm actually talking to you today, and all of that was an introduction, is to a new biomarker that's just been developed, that's just being developed. And it's around a protein called TDP43, or TAR DNA binding protein 43. It was discovered by Virginia Lee and John Trojanowski back in 2005 when the first publications came out. And we know that that protein is normally inside cells, in the nucleus of cells. And those are, and you can't see it well here, but the cartoon on the left shows normally this protein, as shown in green in that slide, is very much in the nucleus. But in disease like ALS, but actually in Alzheimer's and also frontotemporal dementia, it begins to sort of leak out into the cytoplasm, the milieu around a nucleus, and then eventually aggregates. And the histology picture in the middle of the slide, and you can't see it real well, those dark brown dots, that's the aggregated protein. Now we know when it's no longer in the nucleus, it's not doing what it should normally do. It normally should only be in the nucleus and carry out functions relating to the metabolism of DNA called RNA splicing or RNA metabolism. And we know artificially, if we remove it from the nucleus, and that's shown in the far right panel where you see blue circles and yellow circles, the blue circles are all the genes that are not handled properly when that TDP43 is not in the nucleus. This turns out to be, this defect is present in more than 98% of all sporadic ALS patients, all C9 patients, although not in SOD1 patients. Don't understand that yet. I'm not going to talk about that. So it's a very common marker of cellular dysfunction. That's not cell death. And neurofilament's about cell death. Cells are gone, but this is cells that aren't working properly. We know also, and I'm not going to go into it now, that the first thing that happens is you lose TDP4 from the nucleus, and then later it aggregates. But losing from the nucleus, it's called the loss of function, is a very important point. Next slide. There's a little sciencey here. I'll click it one more time. I didn't realize it. The top black bar shows normally when TDP43 is in the nucleus, the DNA is metabolized a certain way. Your DNA is a bit like pearls on a string. We all know the pearls are important. The string's not so important. It holds it together. Well, in our bodies, those pearls are called exons, and the string, the connecting part, are called introns. And TDP43 works on that intron region. Those are those little green circles. Normally, you have to remove the intron, so you push all the exons together. That's how we make proteins in every part of our body. But in TDP43 not working properly, all of that gets mixed up improperly, and you get mixes of partially made exons, and it's improperly arranged. And sometimes those proteins are made, and they're called cryptic exon species. And so they're aberrant proteins telling us that TDP43 is not working properly. And it turns out we have ways of detecting those proteins. So go to the next slide. If you could, thank you. So all of this, just a little background biology, because I don't presume all of you know all the biology that goes on. Every cell has a nucleus that's shown in the left-hand side of that slide. That purple part is the nucleus. Every cell inside the cell, there's a nucleus, and outside the nucleus are all the elements that allow a cell to operate properly. Mitochondria, things called endoplasmic reticulum. But the core of the nucleus and how it communicates from inside the nucleus to the rest of the cell are things called nuclear pores, and they regulate what gets in and out of the nucleus. It's the most complex protein in mammalian, our cells. It's made up of 1,000 proteins that work together in this circular complex. There are about 2,000 nuclear pores for every nucleus. And we learned over the years that there's defects in the nuclear pore in both sporadic ALS as well as familial ALS. And that's the next slide. And this cartoon on the left summarizes what we know happens. What we know is when the nuclear pores not working properly, TDP43, which is normally inside the nucleus, those little orange red circles, it's broken, the nuclear pores not working properly, and TDP43 leaks out, is shown in the lower part of that cartoon on the left. We actually also know there's a process, a pathway by which all of that occurs, or at least most of it occurs, and it's a protein that leaks into the nucleus, damages the nuclear pores, and then later TDP43 leaks out, and that's the whole graph on the right. And I won't go through it other than to say there's an event that occurs in your cells, those of who have ALS, that injures the nuclear pore, and then TDP43 leaks out, and then all the bad problems because TDP43 is not working properly occur. Next slide. To do these kinds of studies, this gets back to what I told you about Answer ALS. We make iPS cells. And by the way, when I say we, again, it's a royal we, not just my lab, but the Answer ALS program made 1,000 of these lines. These are like 1,000 biopsies. And we use these as a surrogate. We can't go into your nervous system and remove that tissue, but we can make these cell lines and determine the defects in patients. Next slide. When we've done that, and this is sort of a busy slide, but if you see the right-hand panel, you see a ton of little red squares, and there's columns. The column next to the yellow column, all those yellow and green columns, those are sporadic ALS patients. This is 150 sporadic ALS patients, only we're looking at their biopsies. All of them have defects in this nuclear pore. That's the first set of red columns on the left. And then all of the subsequent columns, the little mosaic of little more red, some less red, some pink looking, those are measurements of the TDP-43 dysfunction. And this tells us that all of those sporadic ALS patients from that biopsy, if you will, have defects in TDP-43. This is important, but you'll notice they're not all identical. The panel on the left is really the measurements of those various TDP-43 misprocessed RNA species that we now measure in these patient biopsies. Next slide. So we know it occurs in patients, and it's quite variable. Can you click on that again? And one more time. Good, perfect. And what you see is sometimes some of those species match up. They're all the same. Stathmin-2, which is one of the future gene therapies coming up for ALS right now in Canada, but soon coming to the United States, it's very abnormal in sporadic ALS patients, but not all patients. The red bars tell us that some sporadic ALS patients have defects in TDP-43, but not Stathmin. And why is that important? This means that when we start carrying out trials looking for this and drugs that'll fix Stathmin or some of the other TDP-43 misprocessed species, which is the future directions for much of ALS, we're going to need to know, how is TDP-43 mishandled? Which patients have problems with Stathmin? Which patients have problems with other markers? And by the way, independent of that, which drugs are fixing this fundamental core pathway in ALS? To have that, we have to have markers. Next slide. As an example of why that's important, on the left now, you see that same slide of many of those patients with lots of TDP-43 defects. And one of the drugs not yet ready for the clinic, a drug called antisense oligonucleotide to CHIMP7. You just heard, hopefully, about antisense oligonucleotides fixing SOD1 for only SOD1 patients. This shows how well CHIMP7 ASO, that's a short term for antisense oligonucleotide, fixed 20 to 30 of our patients' defects. To know that that's actually working in patients, this is right now biopsies in a culture dish, to know it's working in patients, we have to have a way of actually measuring these TDP-43 misprocessed species in our actual patients. This now is the lead into what becomes the next generation of true functional biomarkers in patients. Next slide. So two labs, Michael Ward at NIH and Phil Wong here at Johns Hopkins, have just begun to develop those really critical new generation of biomarkers for TDP-43 misfunction. When TDP-43 is not working properly, there's potentially many proteins we can measure. This is just the start. This is our first toe in the water. And in this slide, it's busy, but it really says, you can see the middle panel, that when Phil Wong made an antibody to just one of those TDP-43 species, he could find it was markedly abnormal in symptomatic, even pre-symptomatic C9 patients. And not shown here, he also found it in some sporadic, well, actually it is shown here in the red box, sporadic ALS patients as well. This is a very early study of just one of those misprocessed species, but it tells us we can make reagents, tools to measure this. By the way, this is all done in cerebral spinal fluid of our patients. Next slide. Michael Ward, on the other hand, not only looked at just one, he looked at almost a dozen of these different species in a different way. He can also detect these species in patients, in this case in patients' brains at autopsy, but soon we'll be doing it in CSF, again with antibodies. And between both labs, we're seeing the generation of what are going to be the next future of ALS biomarkers, not death biomarkers, but truly functioning biomarkers. By the way, not that there's anything wrong with NFL, but this is our next generation. Think of the first generation as, I don't know, go-karts, and now we're moving into real automobiles in terms of what we'll be able to do. These will be very important in future trials as we move forward in ALS. So next slide. So what's coming now as we look forward the next few years? Multiple TDP-43 readouts. These are going to be antibodies we'll be able to read out right now, really in CSF from patients. Right now, this cannot be done reliably in blood. That's okay. CSF, we can do spinal taps. And actually, as the bottom of the slide says, it's going to require that we do more CSF testing. You know, when I started my career, every ALS patient had a spinal tap. where we measured CSF. We don't do that as much anymore. This is going to be a critical phenomenon that we're going to need to do in our regular clinics so we can have these excellent measures of our drug, how our drugs are working early in the disease. Obviously, late in the disease, we want to see a drug works because patients' disease has been halted. But early in that, and most importantly, validating drugs, we're going to need these markers. So we have a few now. There's a lot more we have to learn. How reliable are they? How sensitive are they? Do they detect, are different markers associated with different stages of the disease? We're going to be able to use some of the samples already stored in, say, in the HEALEY platform trial for some of the trials where CSF is being collected, but we're going to need a lot more that we're going to learn about in the future. So in this very quick, and I apologize that I went so fast, I'm from the East Coast, I talk fast. We're learning about really we're the cutting edge of new biomarkers. And again, should anyone have questions, I'm happy to talk about this either on the phone or through follow-up Zoom calls or through emails. I have to tell you, I type really poorly, so Zoom and emails and phone calls are better. Anyway, I'm happy to take questions through this, and I know we're going to turn over to the rest of the HEALEY discussion.
Thank you so much. This was really an amazing overview, and thank you for sharing, the most important topics in ALS science biomarkers right now, but also very new data, really important and relevant, timely presentation. Thank you. A couple of questions that have come in while you were speaking. One question is, why do we still not have biomarkers such as neurofilament to diagnose ALS?
So, okay, a very important question. Neurofilament changes are seen in any injury to the brain, Alzheimer's, frontotemporal dementia. So it's, you name it, multiple sclerosis, a head bump. All of these things can raise neurofilament. So there's no specificity for disease. It responds to therapy, but it's not specific for ALS. We know the levels tend to be higher in ALS than, say, frontotemporal dementia. I'm picking that one. I can't tell you exactly which one is, but it's also, if I ever show you a graph of it, it's very variable. So right now it's, and I don't think ever in any time in the future, it will be diagnostic.
So this week has been really had a lot of news about ALS, and among those there were a series of presentations. There was many presentations at the MDA, the Muscular Dystrophy Association conference. So I wanted just to start by saying, Jeff, if you have any take-home messages or any important findings that you want to share from that conference or other recent conferences.
I think the most important thing, probably by everyone knows today, is the near approval, in my opinion, of tofersen, which is going to be really the first gene therapy for ALS. We'll hope by April, when the final meeting occurs with the FDA, that they'll approve that. That's going to change things dramatically. And I should point out, and maybe this is getting a little away from your topic, but it's an important discussion that we'll all have as clinicians. As we develop gene therapies that have to be given intrathecally, that is not a normal injection or a pill, all the clinics in the United States that want to do this are going to have to develop special clinics where we actually infuse this. This is a new thing for us. It's been done in spinal muscular atrophy for several years now, but for adults with ALS, this is going to be a new approach, and we're all going to have to work hard to make sure we can get this to patients as quickly as possible. That looms in the future. Scientifically, there's a ton going on. The development of these biomarkers, some new models around ALS, around some of the new genes that have been discovered. The recent Packard meeting, new genes were talked about, new approaches to the disease, interesting information about neurons versus glial cells. So I'm not being very specific because I don't, one, it's others' work, not mine. But I think all in all, this is an extremely encouraging time. And in a grand way also, this used to be, just for those of you who don't know, this used to be a very United States-based research approach. Today, it's truly a collaborative group in Europe, United States, and Asia that have been working together on understanding ALS.
Great. And I see that Catherine is posting a few important notes in the chat. So one is about another webinar that we're hosting in addition to these Thursday webinars. So from the platform trial team, we will be hosting a webinar on Monday, March 27th, about regimen F, which is the regimen that we are starting to enroll for right now in the platform trial. And on the topic that Dr. Rothstein was mentioning, lumbar punctures, infusions, etc., I do want to mention that many of us are working collaboratively again with some colleagues to think about ways to develop resources. And so I would ask Catherine to please post the link to the NEALS Consortium educational webinar webpage. Right now, we don't have one dedicated to person specifically, but I hope that there will be many more educational webinars through the NEALS Consortium that will cover many of these topics. So please continue to check out both the Healey Center website for new webinars, but also the NEALS website. So Jeff, a few questions
I see them.
about, so for example, for somebody who has sporadic ALS, could you speak about glutamate and TDP-43?
Yeah, sure. Since the glutamate hypothesis came from my lab many years ago, there's no direct relationship. Glutamate toxicity is just there are many general events that go on in ALS. Glutamate toxicity, some people think oxidative stress. So it's what we call a downstream event. Doesn't mean it's not important. Really has always been proven multiple times over that it has a reliable, albeit small, effect, and that's an anti-glutamate drug. I can tell you one thing, glutamate toxicity per se does not induce the TDP-43 defect we see. This is a very much more upstream defect. Maybe in the next year or two, we'll have more data how we think this is actually a molecular defect in sporadic ALS as a type of genetic defect, but we don't have enough information. Nevertheless, it's an important target. One day we'll learn whether TDP-43 is even a more important a target.
Yeah. There's a number of questions about TDP-43 actually as a target. So is it relevant only for TDP-43 targeted therapies or cryptic exons and this pathway would be irrelevant for all forms of ALS, many therapies?
Yeah, so fixing TDP-43, one might argue, is almost like fixing the gene defects. And it's a very upstream. We think of a cascade going on. When we see a patient, we don't know where they are in the cascade. TDP-43 is upstream, meaning it happens earlier. And we've actually, for what it's worth, we know that from autopsies, rare autopsies where patients had a brain biopsy years before they developed ALS. The first thing that changes is the loss of TDP-43 from the nucleus, then later it aggregates. So it's important. But once TDP-43 is not working properly, there are many other events that occur in a cell. Both regimen F and regimen E, if I remember the drugs properly, work at some of those downstream pathways. So they equally become important for us to evaluate. Whether those drugs will affect TDP-43 by fixing it later, that's where these biomarkers become important. They'll teach us, are we in some way by fixing say the calico drug, will it fix some of this? We don't yet know, but these will be important as well.
So the
question, please go ahead.
Go ahead. Oh, okay, I was, somebody asked if TDP-43 is so important, why would targeting stathmin-2 only be effective?
So to be fair, as clinician scientists and knowing the science, some of us certainly say the same thing. Is that sufficient? So I showed you that slide where there are hundreds of RNA species that are abnormal, but we don't know the impact of all of those. So stathmin is the most changed RNA. On one hand, that could mean, wow, it's really important. Fixing that one, maybe the other ones don't count that much in terms of the injury to a cell. Could be, and that's every reason, because we know how to fix stathmin right now. We absolutely should do it. Does it mean one day that we'll need to fix 5 others? Sure. The chimp ASO that my lab has been working on, which is not a drug therapy yet, it fixes all of them. Will that be a better drug? Might be, but I don't know. And I'm certainly not going to wait till that one becomes a therapy when stathmin repair might be very important. We know from a paper that just came out that just fixing stathmin in a mouse could be very important in terms of mouse model. Now, some people hand wave about, oh, mice, they haven't taught us a lot. They're very valuable. And it teaches us that stathmin could be valuable. And so I can tell you is in my clinic, when stathmin ASO gets here, I'm going to recommend patients go for it. Will it work? I don't know. I don't know. None of us have that crystal ball for any drug. We get excited when there's good science behind a drug, but beyond that, we have to test them.
Yeah. So tell us more about nuclear pores. How are they formed?
So every organism has nuclear pores. I mean, yeast, bacteria. It's critical because it is think of it as a tennis ball inside your cell. It's got little holes. It regulates how RNAs get out of the nucleus, because RNA is made in the nucleus and then it gets out to make proteins in the cells. It regulates what comes back in. Even its proximity to genes within the nucleus regulates gene activation. So it is a central regulator. If you don't have nuclear pores, okay, you don't live. In ALS, those complexes of thousands of proteins are missing bits and pieces. They're there, but they're missing, and they're not moving things in and out of the nucleus properly. We know there can be mutations of some of those nuclear pores. One discovered many years ago for one of those 1,000 proteins causes a childhood form of motor neuron disease. Another one of those proteins, when mutated, causes a disease that looks like Huntington's disease, and yet completely unrelated. Another one of those proteins, when mutated, causes heart problems. So there are many things we don't know, but there's a lot of very fundamental cell biology about how nuclear pores work. And now we're beginning to apply that knowledge to how they may dysfunction and disease. And fortunately, and I'll end by saying this, fortunately, we know that there are pathways responsible for injuring the pore, and those pathways we can target with drugs. And all we're waiting for now is to find out, are those drugs safe to use? And once we know that, hopefully, the drug companies will move it forward into a clinical trial.
All right. Another question, what kills the motor neurons? Is it missplicing or TDP43 aggregation?
So now we get into science biases. The simple answer is no one knows. I don't believe that aggregated proteins cause a disease. It's garbage that's clumped. Lots of diseases have clumped garbage in the cell. In fact, all of us watching this right now, astrocytes in our cell have little spheres of little stones called corpuscle amylaceae. Yet we're all normal. So aggregates can be associated with disease. I don't think that's the whole answer. Doesn't mean they don't contribute. But what kills a motor neuron, there are a lot of ways. My colleague Bob Brown once had a slide and showed that there were at least 40 different gene mutations that when mutated, all cause a motor neuron injury. So there are a lot of ways in which a cell can die. Just like as human beings, there's a lot of ways in which we eventually become ill. It's not just one pathway. There may be things that are very upstream of a cascade, but by the time we see patients, there may be multiple pathways. And this gets to someone else's question, will we need a cocktail? Yeah, almost certainly we'll need a cocktail, because when we see a patient, we don't know which of those pathways dominate. When I teach medical students and graduate students about this, I have the slide of all the different pathways. And what we don't know, and this gets back to biomarkers, when I see a patient in my clinic, is there a dominant pathway? Is it Stathman in some patients and protein X, Y, and Z in others that are dominating? We can't discern that right now. So we do what's appropriate. We try to treat the ones we know. One day when we understand these biomarkers, it's coming back to CSF and having the right reagents to study from patients, I think we'll have a better way of applying the right drugs and the right cocktails to the right patient. The things we do very well today, or relatively well, in cancers.
Yeah, so to that point, for the new regimens F and G, why is it important that we collect spinal fluid from those regimens?
So for at least two reasons. One, I don't work for those companies, and my guess is they have certain markers that are going to teach them whether their drug is working. Regardless of what I told you about TDP43, these drugs work on certain cellular pathways, and it's extremely important to know if that drug is working that pathway, because if the drug fails, which we don't want to happen, but if it fails, did it fail because it never worked on the pathway they shouldn't think it's happening? Sabrina and I sit on a review committee for NEALS, and it's surprising, unfortunately, how many companies come to us and say, we don't really know how the drug works, or we're not measuring how it's working in patients. And when their drug fails, did it fail because it just never got to the brain? It wasn't the right dose? Or because it failed because it got to the right target, and that target's just a bad target, time to throw it out and move on to new ones. Yeah. That information comes with CSF. Sorry, Sabrina, go ahead.
Oh, no. We just have so many questions. I just want to make sure we get to all of them. Yep. This audience is always very engaged, which is great. So you spoke about new genes. So are the gene panels going to be updated to test new genes? And also, are new genes going to be targeted aside from the ones that we already know about?
Yeah, so a great question. So panels, none of us in academics control what the companies test for. Right now, it's a pretty comprehensive panel. There are new genes being discovered, and honestly, it's up to them. I don't serve as an advisory committee to those companies, but maybe someone from NEALS does, or they just pay attention to the literature. By the way, a lot of these new genes aren't major genes like C9. They're relatively rare genes, but that doesn't mean they shouldn't be tested. So that's one answer. What was the second question, Sabrina? short-term
If they can be
memory.
targeted. I know that, for
Yes.
tofersen, we target SOD1.
Yes.
How about other genes?
Yeah, so each gene has to be, quite frankly, studied individually, whether it's targetable. Sometimes in some genes, you want to turn off the gene. In the grand scheme of things, that's much easier to do with today's drugs. Tofersen, which targets mutated SOD1, we just get rid of SOD1. We stop it being produced. But some proteins, if you turn it off, that could damage a cell. So we have to first find out, is it safe to do that? And today we either do that in human cells, the iPS cells I told you about, or sometimes in mice. The drug I told, the CHIMP7, we already know in mice, if you turn that gene off in mice, that's fine, mouse is okay. But we don't always know that. And some genes, we don't yet know why it's causing toxicity, or some genes we have to make more of it. That's a harder challenge, to make more of a protein when it's bad, if you will. And so it does require often knowing more about, it's not just the mutation, how is that mutation causing disease?
Yeah. So there's a question about antiviral drugs. Could they have a role in ALS, or is this related to what you were discussing?
Yeah, I don't, there's no direct role in what I'm discussing today, and I have to be careful. All of us have a range of expertise, and that would be out of my range, so I won't tackle it. And I know my colleague, Avi Nath, who used to be here at Hopkins, now at NIH, is carrying out some trials looking at antivirals. It's a more challenging approach, because you first want to know, is there a certain class of viral proteins abnormal in patients? And if so, will your drug work at that? And I know Avi at the NIH has been working at that. And it's about probably the most I should say without getting way out of my field.
Yeah, there's a question about a recent announcement, again, that was just made at the MDA conference about a company, Koya, working to activate Tregs and some positive proof of concept studies in a very small study. I don't know if you want to comment on that.
I don't. I would let you comment on that instead.
Okay. Yeah, no, I mean, I think it's, obviously these are very small studies. So
Yeah.
I think, it's as Jeff was saying, when we see companies at the beginning, or when we see small studies, obviously we want to look at the science and the way they design the trials. The reality is that we don't know until we test and retest and look at much larger populations. That's the only way to know if something works or not.
Yeah, I'll add to that, because I've been doing trials well before even riluzole, and small studies don't mean anything until they're big studies. People get excited, and there's no drug that's been approved that came from a small study. That's all I can say. You need to do big studies to know, because of human variability. There's too much variability in trials. So, and the other, I learned this from a group in pharma, small studies with small effects never become big studies with big effects.
Yeah.
So
the good news is that we have many more trials now, and thanks to everyone's participation, we can continue to test and progress from small studies to big studies, and hopefully we'll give you answers. So I just wanted to leave you the last word, Jeff, about sort of, your view of the field, biomarkers, what's next? Yeah,
so this is extremely exciting for us. These new biomarkers, our existing ability to collect CSF from patients, this is going to change if we all work together, and we do as a community, which is great, to very quickly move on these markers, use it to study patient subgroups. We can take advantage of existing patient trials. That's fantastic. These are resources we never had years ago. And I think this will herald a new approach to how we carry out trials, make diagnoses, determine if drugs are effective, and fingers crossed, move drugs faster to patients.
I agree. And I had to say, it's amazing to see how, with this collaboration between the clinical research community and your community of basic scientists, it's amazing to see how we can begin to test all of these biomarkers, basically, or we will soon be able to do that in actual trials. And it's because of this collaboration and kind of discussions that we can bring all your great science to the clinic and kind of work together. So thank you for all you do.
My pleasure.
And we'll definitely have you back on this webinar. It's obviously lots of questions, great questions as always. Thank you very much, and we'll see you next week. Thank you.
All right.